So I'll just start with this cartoon that some of you know, but those who are not familiar with this cartoon, this is, you might remember that from your high school teacher, and showing the chromosomes. And since we are not we are not bacteria anymore, we evolved to eukaryotes and cells that are more developed. Our DNA stopped being circular. That's what happens in bacteria. They have circular DNA like plasmids. And we started having a linear DNA, which which gives a lot of advantage from the evolutionary point of view, especially for exchange of chromosome material and genetic material. But it imposes two important issues to the cell that are very critical. One of them is how to protect the ends of the linear chromosomes. Our DNA repair machinery works very well to recognize the ends of the chromosomes, like the 5' and the 3' end of the DNA molecule, and recognize that as a broken DNA. And every time the DNA repair machinery sees a 5' hydroxyl or a 3' hydroxyl, it binds to these ends and try to mend this linear DNA to try to fix it, and either by homologous, non-homologous end joining or homologous joining of the DNA to fuse the ends of the chromosomes. So this is one important problem that the cell has to solve, that is to hide the natural ends of the chromosomes from the DNA repair machinery. So this would not be mistaken for a broken DNA. And to do that, we have very special structures at the ends of the chromosomes that are the telomeres, that are repetitive TTA-GGG repeats, hundreds to thousands of TTA-GGG repeats at the ends of our linear DNA. And they are covered by a series of proteins that bind to the double-stranded DNA, specifically to the TTA-GGG repeats. And they're collected called sheltering. Also, there is this very specific structure that is the T-loop. So there is a 3' overhang here, so the 3' end strand, it's much longer than the 5' end, so there is just one single strand, and it folds back and intertwines and make a lace at the end of our chromosomes, as a lace that you do in your shoelace, for instance, and it hides the 5' and the 3' ends of the chromosomes from the DNA repair machinery. The other problem that happens when we have linear DNA is that the DNA repair machinery is not able to fully duplicate the end of our chromosomes. So every time the cell divides, the telomeres get shorter. And this is also a fancy way to look at the telomeres, and just to show that using a fish probe, to show that in metaphase analysis, we can see the telomeres at the very end of each chromatid we have. However, if you look closely to this figure, we see that there is a lot of heterogeneity in the telomere length in our chromosomes. So you see that the signal is much brighter in some ends of the chromosomes, like for instance here, than in others here. And even if you look at the same chromosome, the long arm and the short arm have different lengths in the chromosomes. Like even in this chromosome here, the long arms have apparently brighter signals than the short arms. So there's a lot of heterogeneity there. As I was saying, the second big problem of having linear chromosomes is that the DNA polymerase doesn't fill in the 5' end of the chromosome. So every time the newly synthesized DNA is a little bit shorter than the original DNA template. And you can think that if the cell duplicates too much, at the end, they have very short telomeres, a phenomenon that we call telomere shortening. And this is a fundamental process in human biology, or in not only human biology, but eukaryote cell biology, that when the telomeres get too short, it activates the p53, p16, and it activates senescence and apoptosis, and so the cell stops dividing and even go activate cell death. And this is the telomere length, not only in the cell, is a very important marker for cell aging, but if it translates to organisms like ourselves, it also translates into aging of the organisms. And this is an example of telomere length in human peripheral blood cells, more specifically white blood cells. This is from age 0 to about 100 years old. And this is peripheral blood. Each dot is a healthy volunteer peripheral blood telomere length. And this is the length of the telomeres in kilobases that it can measure using different methodologies, or southern blot, that is the gold standard, or qPCR, or flowfish, both that Fernanda Gutierrez here in the lab developed, or using just regular fish, as I showed in the slide before. But we can see that no matter what we do, our telomeres get shorter as we age, and that's the best predictor of aging in a given individual, better than the cholesterol that you measured from time to time. So the cells that have highly proliferative capacity, and you can think of some cells like stem cells or adult stem cells, for instance, hematopoietic stem cells, they have a high proliferative demand. And so they have to counterbalance the telomere erosion with some sort of mechanism so they keep their proliferative capacity. And to do that, our cells evolved to a very unique method to synthesize DNA in our cells, that is by reverse transcription. So we have one single reverse transcriptase in our bodies, that is telomerase, that has a reverse transcriptase motif that is very similar to the reverse transcriptase in, for instance, the HIV virus. In fact, zidovudine or AZT or the reverse transcriptase inhibitors that we use to treat patients with HIV also inhibit telomerase, because these are pretty much the same motifs. And this enzyme uses a RNA template here represented in red as this RNA molecule as a template to extend telomeres. And it has exactly this CCUAA template, and this is why it extends as TTA-GGG in our cells. And the 3' end gets extended by telomerase. And for that matter, DNA polymerase eventually will fill in the gap here. But this is the method for elongating telomeres in cells that have a high proliferative capacity. You can think of that since this regulates a very specific area of our genome, telomerase expression is highly regulated. And the telomerase complex expression also is highly regulated in cells and highly repressed in somatic cells. So the telomerase complex is composed of the reverse transcriptase itself, the RNA template, this carrin that is important, I'll tell you in a minute, and other proteins here that stabilize the complex, as well as TCAP, that is a chaperone that takes it in contact at the nucleoli to get it in contact with the telomeres themselves, that is a substrate for telomerase. So, as I said, when telomeres get too short, we can barely see them here in the cells, and most of the cells with short telomeres will then upregulate P53, P21, 16, and cell goes into senescent apoptosis. Patients with deficiency in telomerase, and the prototype is dyskeratosis congenita, but also some patients with aplastic anemia, that is the bone marrow failure, hematopoietic stem cell failure syndrome. They have mutations in components of the telomerase complex, and they do not they have very short telomeres. In patients, the hematopoietic stem cell is incapable of adequate proliferation and adequately producing peripheral blood cells, and patients develop pancytopenia and an empty bone marrow. That is aplastic anemia. But a minority of patients, because of the telomeres are too short, they eventually evolve with chromosomal instability, and the most common pathway that we can see there is that since the telomeres are not well protected, you'll see end-to-end fusions of the chromosomes, and this will eventually lead during cell division either to aneuploidy or to translocations, and this can cause malignant transformation in these cells. And in fact, patients that I'll show you here with aplastic anemia might evolve to myelodysplasia or AML, and telomere length is the most, probably the molecular mechanism by which these patients evolve to malignancy in the context of an immune disorder. So this is the telomere length using qPCR for healthy volunteers in yellow, and this is the telomere length of patients with aplastic anemia in blue. And if you look at the patients who had short telomeres for their age that are in this lower quartile here, and we look at how patients evolve from diagnosis, from telomere length at diagnosis and over time to immunosuppressive therapy, we see that the rate of relapse, disease relapse or hematologic relapse, is higher as telomeres are shorter. And more importantly, the rate of evolution to myelodysplasia or acute myeloid leukemia is much higher in patients who have short telomeres than in those patients who have longer telomeres, especially monosomy 7, that is way more frequent in patients with shorter telomeres. And this is the most severe clonal evolution in patients with aplastic anemia because it's much harder to treat with either chemotherapy or even take patient in good shape for transplant. And this translates into overall survival, as we see that patients who had shorter telomeres at diagnosis had a worse prognosis over time. So not only short telomeres cause aplastic anemia, but there are other diseases caused by very short telomeres, in addition to aplastic anemia, that are pulmonary fibrosis, and in fact, mutations in these two genes here are the most common genetic susceptibility genes for idiopathic pulmonary fibrosis or familial pulmonary fibrosis, although this is present in only 15 to 20% of cases. And liver cirrhosis also is a clinical manifestation of patients with a telomere diseases. Although I try to convince you guys that short telomeres are a mechanism for bone marrow failure because the cells do not proliferate properly. The hematopoietic stem cells undertake senescence and apoptosis, and the marrow becomes empty. It's much harder to understand why these patients develop pulmonary fibrosis or liver disorder, and especially that they develop a fibrotic disorder in these contexts. So to study these, we developed a biological tool to understand what happens to these patients or to the cells of these patients, either in the bone marrow, the liver, or in the lungs. I won't have time to go into details to each of these models, but I'll just tell you that we use induced pluripotent stem cells to try to produce blood, liver, or lung tissue to study the mechanisms of disease development in these cases, especially because if you go back here, when the patient presents to us or seek doctor, we see that they don't have any hematopoiesis left, or it's very difficult to have any decent lung or liver tissue to study the hepatocytes or the pneumocytes. So we don't have the tissue that is affected is not available anymore for us to study. So you can use the Yamanaka factors to try to reprogram somatic adult cells into a pluripotent state, and then this could potentially be used for therapy, although this is not in the short run, but you can also use that to differentiate these pluripotent stem cells into different tissues. But also we can study telomere dynamics and how telomeres are processed during reprogramming. So we're able to do that, and I'll show some examples here using the Yamanaka factors, Using the Yamanaka factors, we're able to reprogram, and after 21 days, if you're lucky enough, you have these bubbles here that are the colonies with iPS cells. And these are from fibroblasts, but we also do that from peripheral blood erythrocytes quite effectively. These studies I'll show here were being used using retrovirus as the vector to reprogram cells, but more recently we're being using just plasmids for reprogramming, and they work at least as well. So we know that in embryonic stem cells, telomerase is very active, and apparently in the mouse, we knew already that when we reprogram skin fibroblasts into embryonic state or pluripotent state, telomerase goes up. So what we did was to look at telomerase activity in cells from patients with telomerase mutations in short telomeres. So these are the patients here, and there are 4 patients at least with different mutations, but also there is this pair, brother and sister. She had aplastic anemia, and he was clinically healthy. And another patient with the mutation in the RNA component, as well as controls. And what we were able to do to find was that although in healthy controls, when you reprogram cells from skin fibroblasts into a pluripotent state, we can see that telomeres get way longer over passages and over time during reprogramming, as opposed to what happens to here in white that are the parental fibroblasts, that telomeres tend to get shorter. But when reprogram cells from a patient with a telomerase mutation, it's very difficult to get any telomere elongation. So this is a summary of all the graphics for all patients here, which is much easier to see. But we can see here the controls, and when reprogram cells for patients with mutations, either TERT or TERK, you can see that in average, telomere elongation is apparently, or on average, half of that we can see in patients with long telomeres, or with normal telomerase activity. Which is also interesting is that this is the pair of brother and sister. She had aplastic anemia, and she had way less telomere elongation than the brother in all clones, suggesting that there are other genetic or maybe epigenetic events that are contributing to telomere elongation in these cases. More recently, Flavia Donaitis in the lab, she reprogrammed cells from a patient with the DKC1 mutation, that is the discarrying, the gene encodes for discarrying, that is in the X chromosome, and causes very severe dyskeratosis congenita. And as opposed to what we see in the mutations in the telomerase genes, all clones that she derived over time, they got very short telomeres. None of them were able to elongate telomeres as we saw in controls in the previous, slide. In the previous slide, like this one here. But these were able to at least maintain telomere length. But for the patient with DKC1 mutation, you can see that there is a rapid telomere shortening over time in the first passages, at least until passage 10, and then they tend to stabilize. What is interesting is that she has about up to passage 100 right now, and the colonies are still working. So probably there are other mechanisms that are operating here. Some recent results from her suggest that as opposed to what we see in controls, here telomerase is activated later. We only can see telomerase activity at about passage 20, suggesting that you have some additional mechanisms during reprogramming that usually are activated very early during the process, but in the DKC1 mutant, they're not able to activate, but eventually, over passage 20, 15 to 20, when we see the telomere stabilization, we can start seeing telomerase activation. So what she's trying to do now is trying to find the molecular mechanisms that are activating this late-term telomerase activation that are not the CMYC, probably, or SOX or OCT4 mechanisms for telomere elongation. As I said, telomeres get too short over cell division, and this is a very important process during hematopoietic stem cell transplant or bone marrow transplant. So this is a very elegant paper from Lucio Luzzatto in the 90s, because it's a very simple and very effective in terms of the message it translates to. So what Lucio did was he got individuals who had bone marrow transplant 1 year before and got the peripheral blood from the recipient, went back to the original donor, and got at the same time a blood sample from the donor. So basically, the recipient also was 100% donor in terms of the white cells. So pretty much you had the cells from the same person in 2 different individuals after transplant. And so he measured the telomeres in these 2 samples. As you can see, this is in the donor, so there was no stress whatsoever. But when he measured the same cells but in a different person, what he could see is that there was a telomere shortening for almost every individual when you look at the recipients over a year later. So, and this was approximately a 600 to 900 base pair erosion after 1 year in transplant individuals. And what is more interesting is that when he looked at the correlation between the number of cells infused and the telomere shortening that you can see in these individuals, there was an inverse correlation. So the lower the number of cells that were transplanted, the higher the telomere erosion, suggesting that the hematopoietic stem cells that were transplanted, they had to go through more cell divisions to replenish the bone marrow from the recipient to keep up with hematopoiesis, and this extra cell division led to further telomere erosion or some senescence or aging of the marrow. So you can see that if it's losing about 800 base pairs, it's about 5 to 10 years older in terms of telomere loss in comparison to the blood in the original donors. So the transplant process causes some sort of aging in the hematopoietic compartment because of the extra cell division that happens in these cells. And what is the clinical aspect of this? What is the clinical outcome of this telomere shortening? So Régis Delatour at the Hôpital Saint Louis and I, we looked at first a series of patients who underwent sibling allogenic stem cell transplant for a variety of disorders, but mainly they had AML in remission, or they had malignant disorders. And what we saw was that the age-adjusted telomere length, meaning that we're getting rid of the age effect, was a very important risk factor for treatment-related mortality in these patients. So when we look at mortality rate of patients who had short age-adjusted telomere lengths, the patients with short telomeres had a higher cumulative incidence for mortality related to transplant itself in comparison to those who had longer telomeres. So this is the recipient's telomere lengths. And now a recent paper looked at the telomere length in the donors, and apparently it also has some effect, but we didn't find that in our cohort. And now, along with the Régis, we're looking at a new series of about 700 to 800 patients that received an unrelated transplant. And we're looking at the telomere length on the donors and the recipients with Renato Cunha that sent me some of the preliminary and very preliminary data on the telomere length on about 200 pairs of donor-recipient pairs from this cohort, and we were able to see that the telomere length is working okay. The recipients had shorter telomeres in comparison to donors, but we just received, like 1 hour ago, the code for who is who here in terms of who is what disease and what is the outcome. So because we did it blindly, now we're going to link the telomere length with the clinical data and Renato was not fast enough in 1 hour to link out the data so we could present it to you. I hope you're faster next time. But so now we're going to analyze to see whether we can see any effect of telomere length, of the telomere length in the outcome of unrelated transplant. So the other thing I won't have time to talk about today is how we can prevent this. And we're able to see in patients with telomerase mutations. First, at the a trial that was finished at the NIH that I started while I was still there 4 years ago, giving danazol, that is sex hormone, to these patients. And we have enrolled 27 patients with telomerase mutations, and all of them had not only telomere elongation, but also benefit in the trilineage benefit in the three series, blood series, in red cells, neutrophils, and platelets. Over the period they were receiving danazol. And here in Ribeirão Preto, we have a trial using decadurabolin, that is nandrolone, for the same purposes. And we have, so far, I think, 10 patients that have been enrolled. And at least hematologically, they have improved, but we don't have data on telomere length yet. So to summarize the ideas, our telomeres can get shorter for a variety of reasons. First, the one that the most common, the aging. So no matter what we do, and the alternative is not that better, aging is the most important process of telomere shortening in our lives. But also individuals can get shorter telomeres for iatrogenic purposes. For instance, bone marrow transplant that I showed you before, or even chemotherapy. Although we have some data suggesting that chemotherapy does not necessarily shorten telomeres that much, or at least the chemotherapy, non-ablative chemotherapy used to treat lymphoma or breast cancer, colon cancer. Telomeres get fuzzy. You can get very long and very short telomeres 3 or 6 months later, but 1 year later, telomeres go back to normal. Or because you have inherited a mutation in one gene that causes telomerase not to act properly, and this leads to telomere erosion. And from one side, it's causing bone marrow failure, but at the other side, which is more important from the from the pathophysiologic point of view, from cancer biology, it causes end-to-end fusions and balanced translocations. And we see a lot of chromosomal abnormalities in these bone marrows. And this might be a leading cause of malignant transformation, not only in the hematopoietic tissue, but in other tissues. So I'd like to thank the people in my lab. This is sort of an old picture, but I couldn't find the newest one. So we have here Fernanda, who developed very nice methodologies to measure telomere lengths. Barbara, who is pretty much my boss, coordinating the lab. Flavia, who is a PhD student and doing the IPS work. Raquel, who is working on the hepatocyte side and looking at what happens in the liver. And Natalia, who was working with hepatocellular carcinoma and left the lab about a year ago. Diego, who is a physician and works in the clinical part, dealing with all these patients. And there is Vinicius, working with telomere length. He's a medical student working at telomere length in patients who were submitted to chemotherapy. And in particular, I'd like to thank the collaborators, Regis de la Tour and Gérard Saucier at Hôpital Saint-Louis. Thank you very much.